The next speaker is Professor Julio Licinio. He's from Brazil, but now he's at the Sunny Upstate Medical University in New York, where he is professor in departments of psychiatry, pharmacology, and medicine, and he's the senior vice president of the academic and health affairs and the executive dean and dean of the College of Medicine at the same university. Before he went into Sunny Upstate Medical University, he was distinguished professor of psychiatry at the Flinders University in Adelaide, Australia, where he was the director of translational research and the head of the Medical Research Institute of the same university. He has been doing a lot of translational research. He's very well known in this area. He has working with many universities, and he's the founding director of Molecular Psychiatry, Translational Psychiatry, and Pharmacogenomics journals. Welcome.
Thank you, Benny, for the very nice invitation. I'm delighted to be here. The question that Benny asked me to address is this: the future of depression pharmacogenomics. How feasible will it ever be to personalize antidepressant treatment? And interestingly, in 1999, I got the first NIH grant on pharmacogenomics of a mental disorder. In this case was depression. And I was very hopeful that it would solve all the problems and you could identify people based on their genetic profile and then optimize a treatment. And even started a journal on that premise. But I'm not so optimistic anymore. At least towards the end, I'll tell you how I view this at the present time. But I don't think it's that feasible and that immediate. So, let me see here. Okay. So here are my collaborators in Australia and at different institutions. We are just getting our lab set up at Upstate, so it's not fully established there yet, but it's in construction and to be done soon. So there is this quote here from Francis Collins when people ask, like, what's the advantage of, or the practical advantage for the population of having the human genome sequenced? The most glaring thing that comes is pharmacogenetics. I mean, it's going to optimize treatment, and you're going to have, better and more personalized treatments, and that's the big promise. And the one thing is that once you have the person's DNA sequence, you have the person's DNA sequence. You don't have to resequence. It's like an expensive test, but actually not as expensive as other tests, and but it stays there. So the promise of personalized psychiatry or pharmacogenomics is the right to give to each person the right drug at the right dose to the right person at the right time. So when I was at UCLA, for example, I was actually commenting with the wife of the dean there at the time, Jerry Levy, and it turns out that my mother had died of an adverse drug reaction. And then I commented that, and Barbara Levy said, well, Jerry's mother also died of an adverse drug reaction. And so it's in the States, it's estimated that about 100,000 people die a year of adverse drug reactions. So you have to know which drug to give to each person at the right dose and at the right time. Otherwise, either the person doesn't respond or they may die. So I found this cartoon, which I felt was interesting and funky, about the future of personalized medicine. So you have all the buzzwords that people use in this field. They're all there. So, but I'm not going to spend too much time here, just for fun. So one thing that's very striking is this question: why aren't current treatments for depression based on the immense neuroscience discovery of the last 40 years? And I think in great part it's because the disease is very heterogeneous. So we wrote this article so many years ago, almost 20 years ago now, that we put genetic factors, genes of major effect, which I put a small box because then they're not coming up, so they probably don't exist, and most likely a scenario susceptibility genes of small effect, a number of environmental factors, and then different combinations of these things will result in depressive syndromes, which we very deliberately put in the plural. So our key concepts in depression is that obesity is far more intertwined with depression than appreciated. And there are what I call misunderstood candidate systems, including stress-related neuropeptides and immune mediators and the microbiome. And then pharmacogenomics and genomics are a new approach to old problems. So I'll go over this relatively quickly just to give you a general overview. And this stuff is already published, so you don't have to, you can always go back to the original sources. So obesity is a co-occurrence with depression, and it can also be a side effect of antidepressant treatment. So if you're talking about personalizing treatment, you should try to target treatment in a way that's not going to make people have side effects, including obesity, which can be devastating if you are predisposed to diabetes, hypertension, and the complications of metabolic syndrome. So this is stress. So that's the biological response to stress. So that gazelle is either gonna, the whole body is primed to respond to this acute stressor, that's the cheetah, and the gazelle will either die or will, go on a stretch and run. This cheetah gets tired very easily, so runs the fastest running mammal, but gets tired after like a minute. So anyway, this situation was not, mammals did not evolve to deal with the situation 24/7. And when the gazelle is that woman there and the cheetah is her boss behind, then our biology is not made to handle that. So as a result of chronic exposure to stress like that, some people end up depressed and lean, other people end up depressed and obese, and other people, the obesity kind of corrects for the mood, and then they end up non-depressed obese. So obesity has been described since antiquity. However, the rate of obesity is escalating in recent years. And I'm putting a lot of slides here, so I'll run fast, but what I'm going to show next is, for me, the most astonishing thing that I've ever seen in my career. Like from when I left medical school 35 years ago to today, I've never seen anything so remarkable. And I think that we underappreciate and under-address that, particularly in our field, a lot. So, and it's this. So I moved to the United States in 1984. So this survey began in 85. So I saw this from the beginning. And I don't think American society changed that drastically every year, every single year consecutively, the way you're going to see here. So look at the white states in white have no data because the survey initially was not restricted, was restricted to only a few states and then grew to the whole country. And look at the rates of obesity at the bottom, and you'll see that new rates will be coming up that didn't exist initially. And they will escalate drastically. But this is that's not from one decade to another, it's from 1 year to the next. Look at this. Yes. Let me try this.
Try to use the arrows.
Yeah, I'm using the arrows. Nothing. The
Page up.
huh?
Page up.
Page up. Page down. Yes. Okay. So here you see 92, 93, 94, 5, 6, 7. Now a new category. 8, 9, 2000. Another category. 25, 02, 03, 04, 05. 06, another category, more than 30%, 07, 08, 09, and several states, and then 10, and then 11, another category, 35%. That at that point, some states were 34.9, so they already put a new category, but then they really hit 35%, and that's 13, 14, and 15. So nothing that I've ever lived through is this dramatic. And so 1/3 of the population of those states in dark red and even the orange is obese. So the rate of obesity is escalating.
Self-reported.
No, self-reported, which is an underestimate. Nobody, gives additional weight more than what they have. So that's, and women underreport the weight and men overreport the height. So So this is an underestimation. So, so what's happening that has made this go like that? So one thing is that the levels of stress in society are increasing. This is a very complicated survey from the other APA, the American Psychological Association, but essentially it's showing that over a 5-year window that they studied, the rates, most people experienced an increased rate of stress. And so, so how do people cope with chronic stress and depression? They take antidepressants. So here is a rate of prescription in the United Kingdom. From 98 to 2010, it went up a 3.5-fold. And in the US, 11% of Americans aged 12 years and over take antidepressant medication. The rate of antidepressant medication is higher in Australia, so there it's 11% of the entire population. So, so we have these millions of prescriptions, and there is something that we don't think about very much, which is that typically you say, okay, you take this drug, you stop taking the drug, you're back to normal. Maybe not. So I've seen clinically many people who began antidepressants, then they begin to gain weight, and they stop the antidepressants, the weight keeps going. So is there an impact of antidepressant exposure that's independent of current treatment? So we developed animal paradigm. So my wife, Mali Wong, we've been working together for 25 years. She came up with this model that would cause, the group would cause chronic stress to the animal to mimic the effects of depression, then give an antidepressant, which would revert to depression, and then the animals would have neither stress nor antidepressant, and then they would be on a high-fat diet for 300 days. On an obesogenic diet. So, so that's the paradigm. So antidepressant stress, then you stop both, which happens with many people. So they stop the antidepressant, and then they are on a Western obesogenic diet for the 295 days. And so here there are behavioral correlates of the chronic stress, which are similar to depression, and then behavioral correlates of the antidepressant treatment, which shows improvement. And this is very interesting. So these animals here, the only difference is that this group was exposed to fluoxetine for 2 weeks and this group wasn't. So they had the same stress and the same obesogenic diet. But in the exposure to fluoxetine increased the weight significantly. And that's if you look at the whole group of animals. And this experiment was done in Miami and in 2 places in Australia, so it's very reproduced in our hands. Then, this is if you look only at obesity-prone animals. So not all individuals are prone to obesity, the ones that are, this effect is really magnified. And you see this with atypical antipsychotics, you see this with antidepressants. Some people who are susceptible, they gain a lot of weight. And here was even after the drug had been stopped for 300 days. So I'll be talking about leptin. So we thought, could it be leptin? It wasn't. One of the theories for antidepressant effects is that it's stimulating growth factors in the brain. So I thought, well, maybe it's stimulating growth factors all over the body. And IGF-1 is indeed elevated. So we are now measuring growth hormone levels, which is very difficult to see, to measure using external jugular catheterization to see if it's a growth hormone effect. And that's the summary of the ongoing experiments to explore this in greater depth. So the situation here is that you have obesity and major depression, and then you add antidepressants, and then you have all three there. So about half of the people who start antidepressants stop. And you saw how the rate of prescription goes up. So for the rate of prescription to go up, the same number of people who stop, another equivalent group has to start, so then it will stay flat, and more, so the rate goes up. So for the rates to go up, the same numbers that stop have to be replenished in excess. So the population that's exposed to antidepressants, which is not something that we assess usually, is immense. And could this be a contributor to the obesity pandemic that we see today? So just before I left Australia, Zuming Shin did this study on a 4-year follow-up that he I've been looking everywhere to see if I can find this data, and he had it. People who had taken antidepressant and were exposed to Western diet or not. So the ones that were exposed to Western diet here gained much more weight on that same diet if they had been exposed to antidepressants as well. And the difference was not so bad if they had a prudent dietary factor. If they were sedentary, the effect was also magnified, and if they were a smoker, also there was an effect. So essentially, if you have bad life habits and you take antidepressants, the obesogenic effects of that, of those habits, are magnified and potentiated. And so is this a missed opportunity for prevention? Because, as you know, with weight, once you gain, it's very hard to lose, almost impossible. So we do everything that we can to talk to patients and advise them about strictly adhering to a healthy Mediterranean-like style diet when they begin antidepressants to avoid the weight gain that they will have disproportionately if they eat junk food. I don't think we explain that to people, and that's the case. So if you look in the causation of obesity, there are predominance for some people of nutritional factors, so you could be as skinny as you could be if you eat 2,000 extra calories more than what you need every day, you will gain weight. You have some people predominance of psychological environment, and others predominance of genetic factors. And now I'll go on a little tangent here about this, because this shows you the power of pharmacogenetics. So you give the right drug to the right person at the right time, and they respond. And you give the same drug to other people who are not so right, and they don't respond. So leptin has never become like a commercial treatment for obesity, and many trials have been made. It doesn't work as a drug to treat obesity. However, in some cases, it does work in an amazing way. So, as I said before, in depression, we don't have genes of major effect, but for obesity, we do. So those very rare genes, they help us elucidate the biology of the disease, the circuits underlying it. And that gives us an immense handle on understanding the disorder, coming up with therapeutic targets, and moving the field forward. So this was the first hint that there was a substance that would eventually be called leptin. So this is a spontaneous mutation from a paper from 1950 showing the obese, a new mutation in the house mouse. So in 1994, Jeff Friedman's group positionally cloned it and showed that it was a hormone made in fat cells called leptin, for the Greek leptus, theme, that goes to the brain and gives two signals. One, to decrease food intake by both inhibiting orexigenic pathways and stimulating anorectic pathways. So the brain has two different kind of circuits, one to make you stop eating, one to make you eat. And leptin acts on both in ways that would lead you to stop eating, and also increase energy expenditure. So what can we learn from the actions of obese genes of major effect on human brain and cognition? So first of all, these rare mutations, and there are many others, they essentially helped us unravel the biology. So there are many candidate systems in body weight regulation. I'll just mention leptin as one of them because it's the target of this treatment that I pushed. So I have been studying leptin for over 20 years and looking at relationship between leptin and other hormones, sex differences, synchronicity with other hormones, as I said. So this TSH is one line and leptin is the other. You can see that they are very synchronous. And I spent the year of 1998, 1 month in Turkey, studying this family that has a leptin gene mutation. So eventually, I brought them to the US for studies that you'll see. The girl died before she could be brought because leptin gene mutation can be fatal, and she died as we did all the regulatory paperwork. So here's the pedigree of the family. And so I'll skip this slide here. So this is what happened. That's summarized in a video. Let's see if I can play it here. So essentially the patients came to LA, they lost weight, initially because they ate less, so this is food intake, but it's amazing that like 1 year later, they are eating exactly what they were eating at baseline, but they continue to lose weight because energy expenditure keeps going up. So initially they lose weight because they eat less, then they lose weight because they are spending more energy, which are the two effects of leptin. Let's see if this plays.
These three Turkish cousins, Bayram, Zeynep, and Elif, may help you lose weight. Not with a new diet or exercise video, but in the world of medical research. They may hold a genetic key to the elusive mystery of weight loss. Turns out the cousins are ideal research subjects.
For all intents and purposes, it's as if their body never gets a signal that they have enough food. So from when they are born, they begin eating very voraciously, and they eat large amounts of food, and they've become very obese.
Cousins are from a village in Turkey. They flew to Los Angeles to be studied and treated by doctors at UCLA Medical Center for a rare disorder. They are the only known adults in the world with a genetic mutation preventing their bodies from producing the hormone leptin. When given daily leptin injections over 10 months, 313-pound Bayram Donsat lost 146 pounds. Elif Fakili weighed in at 231 pounds. She lost 90. And Zeynep Fakili weighed just shy of 300 pounds. She lost 93.
So every day for the past 10 months, the cousins have injected themselves with small amounts of leptin. They exercise moderately, but haven't restricted their eating. Here, Zeynep and Elif try on their old dresses. The women have each lost 90 pounds. Bayram, with his old pants, has lost almost 150 pounds.
So I think
this is a monumental occasion. The cousins' last weigh-in before returning to Turkey, where they will continue their leptin replacement therapy. Their lives have been changed forever.
So happy, you cannot imagine. They feel very healthy, very happy. They think that they can, have a normal lifespan. And they are really very different from when they first came.
I've never seen a case like this before. And leptin is a well-known protein, but we really haven't known sort of how to apply it in a medical sense. This family is really sort of pure science in that we know we replace it, they lose weight. Now, for other people, the leptin question means, are there other proteins? Are there other enzymes? Will it interact differently? What's the role of insulin? How do the fat cells work? So it's a piece of the puzzle, but we're certainly not done yet.
Much more research must be done to determine if leptin can be an effective treatment for obesity. But for Bayram, the results are unmistakable. When he flew to the U.S. last year, he needed 2 airplane seats. He's leaving in 1. Tracy Potts, NBC News, Los Angeles.
So here is the actual weight from the patient. A is the man, you see he dropped more than half of his body weight. And the women dropped less, but they're also older. And the C is the oldest one. So I don't know if it's an age effect or a gender effect. But they did lose weight substantially. So we are now writing a 15-year of leptin replacement therapy review, but this one is already out there. And one thing which is very interesting is that for most obese people that you see out there, vast majority, they have too much leptin, so they're resistant, centrally resistant to the effects of leptin. And there is this possibility that they are probably not resistant at the periphery to the effects of leptin. But anyway, so they have very high circulating levels of leptin, and giving more doesn't help. So, but as you know, most obese people, they are not hypogonadic. These individuals, they were hypogonadic because leptin is the signal for puberty and for menstruation. So in anorexia nervosa, for example, when the patients lose weight, they stop menstruating, they go into amenorrhea. That's because leptin goes down. Then if leptin goes up, then they menstruate. Then if they lose weight again. So essentially it is leptin that controls, gives a nutritional signal that it's okay for the woman to ovulate and carry a baby. So anyway, so most obese people don't have a reproductive problem, and they have cardiovascular problems. So they have hypertension, triglycerides are up, metabolic syndrome is there. So these patients, they didn't have metabolic syndrome. Blood pressure was normal, and lipids were all normal, but they were hypogonadic. So so if you think that obese people out there, they have the cardiovascular consequence of obesity, and these people don't, maybe the cardiovascular consequence of obesity are mediated by leptin. And so, and I proposed many years back that this hypothesis should be tested. Then Michael Cowley developed a very broad coalition and showed that leptin does indeed mediate the increase in blood pressure associated with obesity, and even identified the very specific neurons and areas of the brain where that's happening. So I told you that girl that saw in the initial picture, she died. But then we discovered another boy. That's the boy. So if you remember the pedigree for this family, if you think of the lowest row of the pedigree, nobody in this family has died of any cause. They don't die young, so they die maybe when they're old. But nobody had accidents or nothing. So except that the 8 people that were born with this obese phenotype, of 12 people born with this obese phenotype, 8 died, only 4 survived. So the odds ratio of death in the absence of leptin is 25.4, and they die of immune deficits because leptin is also a very potent stimulant to the immune system. So the boy. By the time he came to the U.S., he was 5 and weighed 50 kilos, and that's him there. And then he underwent leptin treatment. So that's 1 year after leptin treatment, couple of years, then later and later. And I had to be providing the leptin all this time. For 18 years I've been doing this, until you'll see what's next. And then recently he approached me on Facebook and wanted to be my friend. And then I checked his Facebook picture, which is this. So very different from what you saw before. So leptin, we did functional fMRI studies in collaboration with ED London at UCLA, and we showed that leptin replacement changes the brain response to food cues in the subjects. And so leptin changes the functioning of the brain, and does it also change the structure of the brain. And interestingly enough, during the course of leptin replacement, they gained brain matter in 3 different areas. And we actually did a few experiments that we would bring them back to the US, keep them off leptin for 6 weeks, and study them on, off, and on leptin. And here are the results. So there is actually a remarkable plastic response of the brain to the presence or absence of leptin. So in the absence of leptin, gray matter suffers. So, and that's particularly so in the cerebellum. So the boy, we did very detailed psychometric testing before leptin, 1 week after, and then 1 year later. There was no acute effect for the 1 week, but for the 1 year there were very drastic effects. And these tests are very standardized, so if any one of you does any of these tests, they are very reproducible. If you test your IQ, it's not going to be 110 today and 150 the next day. It changes like 1 or 2 digits, if that. So you can see that on some subscales, but not others, the boy's performance increased dramatically. So we have suggested that leptin has a pro-cognitive effect. And then, interestingly, this group studying the Framingham cohort in the US simply measured fasting leptin levels in the cohort, 700 people, average age of 78. And here are the people with the highest levels of leptin, and this is dementia here. So you can see the highest levels of leptin, the level rate of dementia, and the lowest levels of leptin by 14-year follow-up is a dramatic difference. And here are the 2 intermediary quartiles. So because of a lot of data that's coming together from different sources, leptin is now beginning to be used in clinical trials for Alzheimer's disease. So the conclusions here are that rates of obesity are increasing. It's a major problem in the US. Leptin is a gene of major effect. It regulates cognition related to food. The leptin pathway is crucial for human body weight regulation. There is a whole body of work that I didn't show here because of the time, done by Dr. Shin-Yun Lee from, she used to be at the University of Texas, San Antonio. She just moved to the Medical College of Georgia, showing that leptin has very potent antidepressant effects as well. And so, so we suggest that leptin may have a role in regulating mood and cognition, independent of body weight. So here is that he was the first person identified with this mutation in this family, 19 years later. Then the last batch of leptin that I had to send to Turkey. And this is very interesting because these are the 2 Turkish doctors that work with us. This is a student from Australia that I sent with the leptin. Here are the 3 patients, and here is the boy. But you can see that the patients are very traditional. And here's the Turkish doctor with the leather miniskirt, so that's, you can see a society in transition. And so after 18 years of brokering this, the government of Turkey and the company that owns leptin now finally agreed that the company would provide leptin directly to like a government pharmacy in Turkey to be given to the patient. So I don't have to do this anymore. So so you can see that pharmacogenetics can work. You know, you can give a drug at the right drug to the right people, and it works beautifully. But then you don't give you can give that same drug to the wrong person. It doesn't do anything. So what do we do with depression then, which was supposed to be the topic of my talk? So so we don't have genes of major effect. So without a major lead, how do we even advance the field? So I've suggested that we can systematically examine social and environmental factors, look at candidate systems, and then discover novel clues without the biases of a priori hypothesis. So I told you that I got this first grant from NIH on pharmacogenetics, which was not funded by NIMH. NIMH doesn't like pharmacogenetics because of the idiosyncrasies of different leaderships that they've had. They have not invested. So cancer has invested massively. NHLBI, National Heart, Lung and Blood Institute, NIGMS, National Institute of General Medical Sciences. They've funded people for like 20 years, but not NIMH. They don't believe in pharmacogenetics. So what I've actually been told, coming from very high as you can go within NIMH, is that, oh, we don't want to work on these existing drugs. They are all dirty. They're terrible. So we're going to focus our efforts on coming up with new treatments. But meanwhile, you know, it takes a while to come up with a new treatment, and then these are the drugs that people are taking. Can't we give them like in a better way, in a more targeted way, in a way that minimizes side effects and maximizes efficacy? It would make a huge difference, but it's not been a priority there, as opposed to the National Cancer Institute, where that is a priority. So anyway, so we, NIGMS, so the National Institute of General Medical Sciences, funded a network of centers in the U.S. to study pharmacogenetics. So I was in LA, so mine is the star here in Southern California, and San Diego studied hypertension, Chicago studied cancer. And it's interesting because they put a consortium together, and then once the centers were funded, they tried to, you know, outsource that funding to the institute of that disease. So let's say the hypertension center, they offloaded to Heart, Lung, and Blood, and they paid for the grant. The cancer one, they, you know, transferred to cancer, and cancer eventually paid. San Francisco has one on cigarette smoking, and it's been paid by the Drug Abuse Institute, and NIMH would not pay for ours, so kept being funded by NIGMS. So anyway, so the premise here is that our current treatment is a mess, is a disaster, and I think people 50 years from now, the same way we laugh at the lobotomy, they're going to laugh at how we treat people, because we get people who have a more or less similar clinical presentation, so it's like collections of people with multiple diseases, but that look more or less the same when you just give everybody the same medication. So some people will get toxicity, so some people will... I had a patient actually that at UCLA that I was asked to consult that more than doubled her weight on olanzapine. So whatever the weight was, she was like 50 kilos, she became, 101. So she just slightly doubled her weight on olanzapine and didn't stop being psychotic. So that's this lady here. Toxicity and no benefit. So why, it's a poison, so why are you going to poison a person if they're not going to get better and if they're going to get terrible side effects? Why expose the person to that? It's almost like a criminal act. Some people will benefit, but they are toxic. But then, at least, if that lady gained all the weight but was totally functioning and free of psychosis, you'll say, it's what you prefer. And some people will benefit without toxicity, and some people have no toxicity and no benefit. So it's just like a waste of time and money and effort. So, so pharmacogenomics studies how this relationship between genetic variations and how a body responds to medication. So the idea is that you would get the patient who is a heterogeneous group, and you do DNA profiling and identify people who would have bad side effects, non-responders, good responders, and try to identify them a priori before you expose them to the drug. So here saying the same thing, which I just said to you, but like in a funkier cartoon. So when you look at pharmacogenomics, there are two branches to it. One is looking at pharmacokinetics. So pharmacokinetics is anything to do with drug availability. So what's gonna be drug metabolism, availability, anything that will bring the drug to the tissue of interest. But so yeah, some people metabolize the drug too fast, so you have to give a higher dose. But if they're not going to respond to the drug, you could, drown them in the drug. They're not going to respond anyway. But if you don't adjust the dose, at least they don't even get the exposure that they need. And other people metabolize the drug too slowly, so they're going to have too much exposure. They're going to have side effects. So pharmacokinetics optimize the dosage, and pharmacodynamics optimizes who should the drug be given to. So here, anything that's happening before the cell here is a pharmacokinetics. And the moment that the drug touches the cell, like a receptor or anything that happens after that, then it's pharmacodynamics. So when will we stop guessing? So this is a cartoon that I got as an example for carbamazepine, which is a drug that we use in psychiatry. Can be highly effective in some people, ineffective in others, and can have serious side effects. And genetic testing can help with diagnosis, with treatment, with pharmacogenetics. That's what we're talking about here, the orange thing there. The yellow one could also be markers of a prognosis. And could be drug development too, because if you identify a gene that's involved in drug response, one thing that's almost like a holy grail of depression is that, as you all know, if you give antidepressants, you need to give them for weeks before the person responds. So it's not the effect on serotonin transporter that's immediately changing the mood, because it happens within minutes, actually, it happens within an hour. So it's something that's happening down the road. So something is changing in the cells down the road. It's a protein, a receptor, a second messenger. So if you find what that target is, you could develop a drug that would go straight there, and then you'd not have to wait 3 weeks. So it could be a very useful target for drug development, but everybody's been looking for that. Molecule has not been yet identified, including we've looked a lot but didn't find it. So another thing is that people take combinations of medications, which is a horrible thing, and I think out of desperation, and was explained in the previous talk very elegantly, people mix different antipsychotics and different antidepressants without any clear proof that they work better in this combination. So Helen Lavretsky is a colleague who is the head of outpatient psychopharmacology at UCLA, geriatrics, and I had geriatric patients. She did a survey in the clinic a while back when I was there. The average patient was, each patient was on average on 17 drugs. And as you know, when you put a drug on the market, you do all the research on that drug. So you give that drug to an animal, you give that drug to a person, you measure, all the different phases, 1, 2, to B, 3, and 4, etc., of that drug. Nobody studies the combinations. So these combinations are not tested in an animal. So you've never given, those 17 drugs to a rat, and then you give to, a geriatric patient and hope that they're going to get better. So, so here we are. So we did this study in LA, funded by NIGMS, and we've been using this protocol ever since. So the phenotype is not depression, it's antidepressant treatment response. And then you have genotype candidate systems searching for new targets, and clinical translation and ethical implications. So this was a double-blind placebo lead-in of 2 drugs, desipramine and fluoxetine. And one thing which is interesting is that we did this I set up a clinic with the money that I got from NIH, but we did 300 patients ourselves. And you see these other studies that, huge multi-center studies with 700 people, but in 20 sites. So if I had 20 sites multiplied by 300, you can see how many patients you could have. So I think the yield is relatively low. Another thing that I think this field has not evolved is that pharmacogenomics, the pharma is pharmacology. Pharmacology is a science. To do a science, you have to give things in a standard way. So you don't do a test tube experiment to just throw some reagents there and then mix and see what happens. You measure very precisely. So pharmacology, you give a drug at a dose and you escalate the dose and you have a protocol and you go. So what people do because there is no funding, people just collect samples from like a clinic and say, oh, we treated these patients with depression, and then some were treated with this, some were treated with that, some were treated with the next thing, and then some got better and some didn't get better. And then let's do a genetics of this. But the pharmacology is very dirty. If you were think about this, if you could not publish the pharmacology by itself as a paper, it's not because you throw genetics that then it's going to become a high-end paper. But you see papers like in Nature Genetics with the pharmacology that would not come up in any, throwaway journal. So you need the pharmacology, but the pharmacology is expensive because you have to diagnose the people, you have to treat them, you have to have a standardized double-blind protocol, you have to see people every week, you have to collect rating scales and see how they evolve over time. So that's very expensive, and it's not testing new hypotheses, it's not sexy, but it's what people need because they need optimized treatment. But funding agencies don't fund that. At least not for psychiatry, they fund for cancer. So the phenotype is antidepressant treatment response, and you can see here that the HAM-D goes down to some people towards total remission, some people are still symptomatic, so the idea is that this difference here could be due to genetics. So we looked at specific candidates, and that's a while back, so I'm giving like a history of how we developed this. So initially we looked at specific candidates, including the CRH1, which had been cloned 10 years before we studied it. And everybody was looking at CRH, and nobody had the idea, okay, maybe let's look at the receptor. So which we did, had a finding, and then once you publish anything, then everybody goes and tries to publish, follows. So we've had over 50 association studies. Then we developed, which at the time was very innovative, bioinformatics approach to SNP identification along pathways. So we would identify a biological pathway and then look at all the SNPs in that pathway. So, and then we looked at multiple pathways, separately. So one of them was this stress/immune axis. That's depicted here. So we looked at brain cytokines in depression through molecular neurobiology, animal models, clinical research, and then genetics and pharmacogenetics. So when I began to look in this field, and the cytokines were first cloned in the 80s, it's obvious that in animals they cause effects that are similar to depression. So I immediately hypothesized that they could be involved in the biology of mood disorders. And at the time, it was not known that they were made in the brain. So there is a hormone that I used to study called cholecystokinin, which is very well known because it's a gastric hormone, but then the brain, once cholecystokinin is made in the periphery, then the brain makes its own cholecystokinin. So it doesn't have to cross the blood-brain barrier. So a signal from the periphery goes to a nervous system, a signal goes to the brain, and then the brain makes its own. So I hypothesized then in the 80s and 90s that that same thing could be happening with the cytokines because they're not entering the brain unless you have serious problems with your blood-brain barrier. So we did a number of papers showing that yes, indeed, when there is an inflammatory stress stimulus, the brain makes its own cytokines. So it's very well accepted now, but it was very new at the time. Then we measure cytokines in the circulation. So this is IL-6 in depression, showing that it's elevated across the 24-hour period and is a disrupted circadian rhythm. This is, then we began to look at genetics and pharmacogenetics. And looking at, we looked at the pathway and SNPs along this inflammatory pathway. And we had some findings on Tibet and proteasome. And given our findings on Tibet, a group from Korea studied the behavior of Tibet knockout animals that were chronically stressed, and they were resistant to depression. So let's say here, when you have forced swimming in the depressed animals after chronic stress, there is immobility, which the Tibet knockout don't have. In the wild-type animals, once you have chronic stress, you have high IL-6, similar to depression. You don't have that in the Tibet knockout, and so forth. So this gave us a hint that these genes could be involved in the biology of depression. So what we've been doing now is looking at whole genome sequencing data, and we had a very brilliant early career researcher, Cheng-Long Yu, he's Chinese, he was in the US, we recruited him to Australia, then he went to Australia, and then we came to the US. So anyway, he's developing mathematical models to sort out people based on their entire DNA sequence and see if you can identify depression and non-depression, and response and non-response. So, so here he's able to separate major depression controls and depressed from a Mexican American population from LA, but from an Australian population he cannot do it. And the reason is that Mexican Americans have twice as many rare variants as Caucasian population. So so you cannot do the same analysis without the rare variants there. So so he published a few papers on identifying depression and diagnosing depression based on genomic sequencing and aggregating kind of regions of interest in one metric that he created. Then Mali also developed a new tree-based approach to looking at multiple genes. So this is not a polygenic gene score, it's a tree-based analysis. So you look at one polymorphism in one gene and then look at the other gene, and then if you have that, you look at another and another and another. You can also add environmental factors. So this was validated in our initial cohort in Australia and in a much larger cohort that we got from the US. And then she had an HMRC grant from Australia to look at that in greater depth. And then in our study, actually fluoxetine was better than the dazipramine over time. And we did a pharmacogenetic study that we had a very interesting finding that's restricted to this population. And you can see the peak here. And it's a brain methylated DNA immunoprecipitation sequencing site, suggesting something that could be epigenetically regulated. And we have actually other findings now that we are currently exploring, but the sample is small. And we also identified this gene, which had been previously identified as a tumor suppressor gene, as being involved in depression. And it is expressed in the brain. It's downregulated by stress, and now we have the knockout animal and looking at the behavior. So rather than present the actual data on depression, pharmacogenetics, there have been a number of reviews that have come up in the last 2 years, some longer. So Alessandro Serretti was a pioneer in this area, and so he has multiple very comprehensive papers. This one is one of them. So he also did an economic analysis if pharmacogenetics should be incorporated in major depression treatment in Europe, both in, his conclusion was that in the high-income countries, yes, in the middle-income countries, not so much. And then another review by Boozman. This was from Mark Fry and Rich Winchelbaum, just showing that from the FDA's perspective, the number of drugs that have a potential pharmacogenetics targets that can be tested and have been submitted to the FDA. So this is FDA data. You can see the psychiatry is actually at the top there. Cancer is not here. Cancer is the number one by far, and then the other, this is the rest of medicine. So, and you can see that the studies, this is 18 from Pharmacogenomics Journal, this is also from 18, and this is from 17 from Spain. This, I think, is very interesting because it's looking at the polygenic risk approach, which is different from what I showed you before, but look at the number of authors, and it's probably more than the number of references. And what I find astonishing is this size, so like STAR-D, which was this whole national study. It's used a lot in pharmacogenetics, but it's very wrong to use it for pharmacogenetics because I really believe that if you're doing research, you have to you have to have a question, and then you develop a method, and then you test it, and then you go. You cannot say halfway through, oh, by the way, I should have done this, and then you do in the middle of the study. It doesn't go very well. So STAR-D was designed without any thought to pharmacogenetics. So after more than a month that the study had been going on, people said, oh, let's collect blood for genetic studies. And they did at that point. But so by the time that the blood was collected, after a month, the study, many patients had already dropped out. So you missed all the adverse reactions, all the dropouts, everything that happened early was totally missed because they don't have blood from those patients. So then you have GenDep, which is a multi-center study, a large multi-center study in Europe, 736 patients, and in our clinic was 300. So you see that, and then you have these other ones. For contemporary genetics, these numbers are pitiful, you cannot do very much. And for the polygenic risk score, there was no finding. So switching gears a little bit, you see all the challenges of finding the genes. Yeah, we have done studies in microbiome to see the other genome. More than half of your DNA is actually outside of your cells. So the microbiome contributes substantially to behavior and to health. So could depression be caused by the DNA that's outside of our DNA, by the bugs? And this could be a wild fantasy, but remember that after decades of therapy and surgery for ulcers caused by stress, it was shown that they're actually a bug that was causing them. So could something this drastic be happening? So there is what's called the gut-brain axis, which includes the microbiome, and it's we had the review in molecular psychiatry that's here. We have two papers that I'm not going to show the data because of time, but essentially showing that if you change the microbiome, you change the behavior of the animals. So these colleagues of ours from China, they have germ-free animals. So if you give microbiome, so they have no microbiome at all. So if you give microbiome from depressed patients, the animals behave depressed. If you give microbiome from normal individuals, healthy individuals, they don't behave depressed. So the behavior is determined by the microbiome and what they receive. And so that's this initial paper here. And you see that the microbiome of depression and control is different, and then when you give it to the animals, they behave differently. And then we did the opposite. We changed the behavior of the animals, both intact animals and animals with caspase-1 knockout, which they are resistant to depression. And then we show that if you change the behavior, you also change the microbiome. So that's that's what this study summarizes. So essentially, you have I have it here. Yes, you have this gut-brain axis in which the behavior is changing the microbes, and the microbes are changing the behavior. And this is a very important potential new target for antidepressant treatment. And interestingly enough, Paul Greengard published very similar findings with animals being resistant to stress by studying a protein called P11. And then we developed a collaboration with them to see if, because they had almost identical results that we had with caspase knockout. And then we are showing that the caspase-1 and P11 interact. So that's what we are studying now. I'm going to skip this here, but the one missing piece that's not in my own talk and in my own work, but something that I want to be doing soon, is looking at iPSC cells as a model for a drug response. So you can take fibroblasts from a person, transform that into a pluripotent stem cell, and then grow that cell into neurons, including serotonin neurons, and then you could treat those neurons with drugs and see if they will respond or not, and that could be a guide to the person. So to summarize my talk here, I was kind of pessimistic about the gene sequence alone is not going to be enough for us to optimize personalized treatment. So Rusty Gage from the Salk Institute, his group, Shani Stern and Krishna Vadadari, whom I know very well, they published this last month. Yes, and what you see here in this cartoon at the bottom is the typical premise of pharmacogenetics. So you take the blood, you look at the sequence, and then you optimize treatment. And they say this is not really going to go like that. So for psychiatry, what they suggested is this. So this is their original figure. So you have of course, their work, so the iPSC cells. Some people optimize treatment using EEG. Then other people do it using brain imaging, and I worked with a colleague in Australia to try to merge brain imaging and genetics. Was very hard. Then here you have the clinical data. Then this is genetics, this is gene expression, and this is epigenetics. So I threw a couple of things here: stress, nutrition, and the microbiome, which they didn't even put there. So I think that if you bring all of these things together, yes, you could optimize treatment. And but who can do this? Who has the brain power to do it? I certainly don't. So I spent a day the other day at IBM Watson, and they have this program called Watson Healthcare to use artificial intelligence to guide treatment. So this is their website, and I have no interest in them. I don't collaborate with them. I don't get a penny from them. So this is not like propaganda from me. So this is what they are doing for oncology, which I think is what we need to do for psychiatry. So that you see there, each person's cancer is unique. The treatment journey should be true. So I think each person's depression is unique. So each treatment journey should be true. So if you go back to this slide here, for cancer right now, they go from here, from imaging, clinical data, genetics, and gene expression. So they go this portion here, and they integrate everything through artificial intelligence, and they develop individualized treatment programs based on that. So I'll skip this here because the last slide, I think this is too wordy. I made something a little more, well, I'll leave it like this. So the way that I think about psychiatry now is that you have the brain with defined functional changes like the plaques and tangles of Alzheimer's, or to be discovered or confirmed microstructural functional changes that alter key functions like mood, memory, cognition, food intake, as you saw. And then how can we approach this? We can go through vertical integration, looking at cells, animal models, first-in-human studies, clinical trials, new treatments, and improved outcomes. Or we can go across diseases, looking at shared biological processes like stress, inflammation, which is important for mood disorders, for schizophrenia as well, microbiome, neuroendocrine changes, or technical approaches like artificial intelligence, induced stem cells, imaging, and genetics. So to answer the question that Benny asked me to address here, this is my summary. So I think that our brains will never be able to integrate all these platforms and provide treatments that are as unique as each person's depression. But I predict, someone said that, in 10 years, what we're going to have in 10 years, in 20 years 10 years was you, right? And 20 years was you. And then I'm 30. So I think by Benny's 100, on the 5th of August of 48, we will have computer-driven artificial intelligence that will, by that time, will have risen to the occasion and will do what our limited human brains cannot process, which is to mine big data and integrate knowledge across domains to provide the psychiatrists and the patients of the future fully personalized treatments for major depression. So we have 3 more symposia set.
We have time for 2 questions.
Outstanding presentation. May I ask a short question? In this Turkish family with leptin deficiency,
Yes.
did you perform some depression or anxiety scores before and after leptin treatment?
I did, and it's the most interesting thing because I did a number of scales, unusual scales for anxiety, for depression, for this, for that. They didn't score very high before or after, but their behavior changed completely. But in a way that you could not capture. I actually have a film of the boy before and after, and you can see the difference. But before leptin, the nurse used to call them our little angels. They were very nice and polite. Oh, lunch is late, that's no problem, whenever, we are fine. Once they got leptin, within days, like the same day, the next day, they are aggressive and assertive. Where is this? I want that. And totally different behavior, but nothing that can be captured in our psychiatric scales. Yes.
I totally agree about maybe the environmental factors and the addition of but thinking a bit practically, do you think we can actually add all this for each and every single patient? We can gather all this information for the predictions, because I understand, you can use the clinical files, you can use genomic data maybe, but what about all these other things, especially
That's why I took the time frame a little longer, because I think I was very impressed with this IBM Watson Oncology, that for cancer they do, and interestingly, because you do a lot of tests that people have that they are just lost in the system. So if you put everybody's As day goes by, I think it's unlikely that anybody has early onset, someone's beginning to get psychotic and they come to a hospital, they have some kind of brain imaging. I mean, unless you're in a very, remote place, I don't think that anybody doesn't do a brain scan of some sort. And then at some point you'll have the whole genome of the person available. And then you have clinical variables actually very predictive. So you have a lot of clinical variables that are not used in any kind of a treatment optimization. So I think that if you put everything that you have together, certainly now, but let's say if you think 10 years from now, 20 years from now, you have a lot of data, and if you put it all together, you may be able to personalize things better than what we do now. And that's what they do for cancer right at this moment.
We have to stop here. Thank you.
Sure.